Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine if we could not just, understand the incredibly complex dance of molecules inside our cells, but actually rewire it like a cellular engineer. For decades, that seemed like, well, science fiction, really, a distant dream.
Absolutely.
But now, a new era is dawning. Artificial intelligence is moving beyond just predicting static protein shapes. Today on the Deep Dive, we're exploring how AI is actively rewiring the very language of life's interactions, designing molecules that could literally change how cells talk to each other.
And our deep dive today, it's into this really fascinating essay from Science Journal. It's titled AI to Rewire Life's Interactome. Structural Foundation Models Help to Elucidate and Reprogram Molecular Biology by Qiao. It lays out this really profound vision for how AI is poised to transform how we understand and maybe even manipulate biological systems.
Right, so our mission today is to unpack what this actually means, how these groundbreaking AI tools work, and why people are calling them a computational microscope for molecular biology. Okay, so let's set the stage a bit. Why are molecular interactions so fundamental in biology? What's actually going on down there?
Well, they're everything, really. Biomolecular interactions are the orchestra conductors of life.
Okay.
Whether it's proteins interacting with other proteins or maybe with smaller molecules like drugs, these precise handshakes, embraces, sometimes even pushes, they allow living systems to transmit signals, generate energy, orchestrate all these fundamental processes. They define the larger organizations we see in biology.
So
Without them, cells couldn't grow, they couldn't divide or respond to their environment. I mean, life as we know it just wouldn't exist.
and for decades, how do we even try to understand these incredibly tiny, intricate dances? It sounds almost impossible.
It was incredibly challenging. For a long time, our main tools were experimental methods, things like X-ray crystallography and cryo-electron microscopy.
Right, I've heard of those.
Yeah, and they're incredibly powerful for figuring out molecular structures, basically giving us the 3D coordinates of individual atoms, like a blueprint.
But there's always a but.
Well, the catch is they are notoriously slow and laborious. You're often talking months of intense lab work just to get a few static snapshots, a molecule in just one specific state.
So like trying to understand a complex ballet by looking at just one single photograph.
Exactly. You miss all the movement, the dynamics, the interactions.
Then came the AI revolution, specifically AlphaFold 2. What was the big shift there?
Oh, AlphaFold 2 was absolutely a game changer, no question. It showed that AI could predict the static, folded shape of a single protein with atomic accuracy, almost as good as the experimental methods, sometimes even better for certain things.
How did it do that?
Basically by learning patterns from millions of years of evolution encoded in protein sequences, and also learning from the structures we did already have from experiments. It was astonishing because it could tell you what an isolated protein looked like. But here's the crucial point, and this is really where this new work picks up.
Right.
Biology hinges on interactions, not just isolated structures. AlphaFold predicted the shape of one protein. This new work is about understanding how things interact, and even more powerfully, how we might change those interactions.
Okay, so that brings us to this computational microscope idea for the structural interactome, that whole network of interactions. What's this new AI strategy called, and how does it go beyond what AlphaFold could do?
So the core innovation here is a generative AI strategy called NeuralPLexer.
NeuralPLexer, okay.
You see, traditional approaches for predicting how, say, a protein binds to a drug molecule, they often assume the protein was rigid, like the fixed lock in a specific key.
Right, the old lock and key model.
Exactly, which simplifies the computation, sure, but it's just not how biology works in reality. Biomolecules are highly dynamic, constantly changing shape, twisting, flexing. NeuralPLexer is specifically designed to capture that dynamic nature.
How does it actually handle that dynamism? The article mentions generative diffusion and one-shot generation. That sounds quite technical. Can you break it down a bit?
Yeah, let's try. Think of it maybe like an AI artist.
Okay.
Instead of starting from zero with countless guesses for every possible shape a molecule could take, NeuralPLexer uses these generative diffusion models. Imagine the AI starts with a really rough, kind of blurry sketch of the whole molecular complex, say, a protein and a small molecule binding. Then it progressively refines that sketch, adding more and more detail, more clarity. But crucially, it doesn't just give you one static picture. It generates an ensemble, a whole collection of different conformational snapshots simultaneously.
Ah, so a range of possible shapes.
Exactly. It gives you a much fuller picture of the molecule's dynamic behavior, all in what they call one shot. This effectively bypasses those really slow sampling barriers that limited the older methods.
So it's like getting an entire animation of the molecular dance instead of just one still frame.
That's a great analogy, yes. You see the movement, the flexibility.
So by giving us that full animated sequence rather than just static pictures, what kind of new questions can researchers actually ask now? What's the fundamental insight this new view really unlocks?
Well, that's the key insight, isn't it? By generating this ensemble of dynamic snapshots all at once, we're not just guessing about static poses anymore. We're seeing the full, flexible choreography of how molecules interact, which is precisely how biology actually works.
Right.
So this allows us to understand not just if molecules bind, but how they bind, the subtle shape changes involved, and importantly, how that whole process affects their function downstream.
That really does sound like a massive leap forward. How did they check it was actually working? How do you validate something like this?
Good question. For initial validation, they use NeuralPLexer to predict the formation of something called cryptic pockets.
Cryptic pockets.
Yeah, these are fascinating binding sites on a protein that are basically hidden. They only get revealed when a ligand, the binding molecule, actually comes along and binds. The model successfully generated these dynamic conformational distributions that were remarkably consistent with actual experimental results. And what's more, it even showed it could distinguish strong binders from weak ones across many different targets, even though it was never explicitly trained on binding strength data.
Wow. Okay. That's a pretty strong validation then.
It certainly is. It showed the model wasn't just making things up, it was capturing real biophysical properties.
So beyond just predicting structures and pockets, how did this AI tool maybe go a step further? Did it provide new insights into how these molecular systems actually operate? Did it reveal something we couldn't see before?
Yes, it absolutely did. The model gave some really valuable mechanistic insights into how specific proteins function. For instance, there's an enzyme called ketyl acid reductosumerase. NeuralPLexer accurately captured the subtle closing motion of a part of the protein when its partners bind a dynamic movement that's crucial for how the enzyme works.
Okay.
And for another example, a G protein-coupled receptor, a really important class of drug targets.
Oh yeah, loads of drugs target those.
Exactly. The model generated a conformational hypothesis, a potential shape, that actually explained why this particular receptor was constantly active, even when its normal signaling molecule wasn't there. So these kinds of capabilities really highlight NeuralPLexer's potential for unraveling complex molecular mechanisms, things like allosteric regulation.
Where binding in one place affects activity somewhere else entirely.
Precisely. And also things like enzyme catalysis, the actual chemical reactions proteins perform.
That's incredible. So it's not just seeing the shapes, but starting to explain the how and why behind the protein's function, those subtle mechanics. But beyond the mechanics, understanding the why often comes down to energy, right? The forces involved. Did they look at that too?
They did, and that's a crucial piece. To complement NeuralPLexer's structural insights, the researchers also developed another related approach. It's called OrbNet-Equi. Yeah, another geometric deep learning method, but this one does focus specifically on studying the energetics of molecular interactions. So this is about understanding the subtle energy changes when molecules interact, down to things like protonation states, electron configurations, really fine details.
Which sounds computationally heavy.
Traditionally, yes. You'd use full precision quantum mechanical methods, incredibly accurate but incredibly slow. But what's astonishing here is that OrbNet-Equi achieves comparable accuracy, but it's about 1,000 times faster.
1,000 times.
1,000 times faster, which just opens up the possibility of looking at these subtle yet crucial energetic changes much more routinely.
Okay, but then they took this whole computational microscope idea even further. From observing and understanding to actually designing entirely new molecules and interactions. That really does sound like science fiction, actively programming life.
This is really where the rewiring concept comes into sharp focus. They enhance NeuralPLexer with what they call an in-painting-based approach.
In painting, like in photo editing.
Kind of. Think of an AI image tool where you select a region and the AI paints or restyles it based on the context around it.
Okay.
Here, the method tries to discover new pockets, potential binding sites we might not even know exist on a protein, and then simultaneously it designs ligands, small molecules, to fit into them. It infers the ligand structure, parts of the protein sequence, even variations in the pocket shape, using only the backbone structure of the target protein as a starting point.
So it's not just filling in a blank. It's creatively proposing entirely new molecular pieces that fit and could do something specific.
Exactly. It's generative design in a really powerful way.
So what does this mean practically? Did it actually work? Was it successful in designing these new interactions?
Yes, absolutely. This strategy led to some really accurate predictions, even on notoriously challenging targets like ARS, G12C.
KRAS, the famous cancer target.
Yeah.
Very hard to drug.
Exactly. And this AI approach achieved a markedly higher design success rate compared to traditional computational design algorithms. And that's incredibly significant. It really opens doors for designing ligands for previously uncharacterized binding sites. It allows us, essentially, to program new molecular interactions.
So instead of just finding existing keys for existing locks,
We can now design new keys for locks we didn't even know were there. Or maybe even design entirely new locks and keys to interact in completely novel ways.
this feels like more than just a new tool. The article positions this work almost as a manifesto, aligning structural bioinformatics with large language models. What's the bigger picture here? How does this shift how we think about biology?
Yeah, I think manifesto is a good word for it. It signals a really profound shift. At its heart, the motivation is twofold. Biologically, it's about deciphering that uncharted, dark interactome, all those molecular interactions we don't fully understand yet, maybe because they're too transient or complex for older methods.
The hidden connections?
And then, translationally, it's about using that knowledge, building new molecules, small proteins, peptides that can actually reroute problematic, disease-causing interactions for therapeutic benefit.
Reroute them.
Yeah, it basically frames interactome mapping almost like a natural language translation task.
Okay, how so?
Well, think about how large language models learn the grammar and meaning behind human words to translate between languages. These AI models are kind of learning the grammar of molecular interactions, how amino acid sequences, the words of proteins, translate into specific 3D structures and binding interfaces, the meaning or phrases of life.
That's a powerful analogy.
And it's critical because traditional methods often just hit a wall. There isn't enough experimental data for vast swaths of the interactome. These AI models can potentially generalize from the data we do have.
And this leads to what they call a closed AI wet lab cycle. What's the ultimate goal there? It sounds like this could really accelerate discovery.
Exactly. It's about creating a continuous feedback loop. Key innovations here include techniques like masked interface pre-training, which helps the AI learn how different parts of molecules, even distant parts, have co-evolved to fit together perfectly, and multi-chain attention to understand how multiple molecules interact within a complex.
Okay.
So the closed AI wet lab cycle means that the AI predicts potential complexes or designs new binders. Those predictions then directly seed experiments in the actual lab, the wet lab.
And the results from the lab.
Feed right back into the AI model, refining it, making it better. It's continuous learning and improvement.
And has this cycle shown results already?
Yes, they mention leaps in predicting context for transient complexes, interactions that are hard to catch experimentally, and also improved success rates in Dovo degrader design.
Designing molecules to destroy specific unwanted proteins.
Exactly. So the ultimate goal is to extend these capabilities even further to more sophisticated interactions, like protein interactions that are stabilized by chemical modulators, something called induced proximity.
Induced proximity, like
Yeah.
using a third molecule as a sort of molecular glue.
Precisely. Forcing two proteins together that might not normally interact strongly. This really paves the way for rationally designing ligands to reprogram entire protein interaction networks. And ultimately, the hope is to restore healthy cellular states.
So we're really talking about therapeutic precision here. Selectively targeting the problematic proteins or interactions while hopefully sparing the essential ones. That could completely change how we approach many diseases.
That's the promise. These emerging capabilities mean we're moving towards designing stabilizers for specific protein interfaces, controlling where pathogenic proteins end up inside a cell, biasing the formation of certain molecular assemblies, all to improve the therapeutic window.
Better efficacy, fewer side effects.
That's the ideal, yes. Making treatments more effective and safer by being incredibly specific. The paper even forecasts, perhaps optimistically but perhaps not, FDA-approved AI-designed interface therapeutics within 5 years.
5 years. That would be a genuine paradigm shift in drug discovery, moving from often serendipitous discovery or brute force screening to rational AI-driven design.
It really would be.
It's fascinating how this kind of complex cutting-edge research actually happens. It's clearly not just one lab working in isolation.
No, absolutely not. This work really synthesizes efforts from major players. You see names like DeepMind, EMBL, EB, various biotech startups involved. It showcases this growing ecosystem where open-sourced models from academia or big tech meet proprietary high-throughput experimental assays in industry. It's a testament to collaborative science pushing the boundaries.
Okay, so just to recap for everyone listening, we've journeyed from AI predicting single static protein structures like AlphaFold did brilliantly to this new generation of structural foundation models like NeuralPLexer that can dynamically predict entire molecular interaction landscapes. The whole dance.
Right.
And perhaps even more profoundly, we're now getting to a point where AI can help design entirely new molecules to intentionally rewire those interactions, intentionally for therapeutic purposes, moving from just understanding to actively engineering life's fundamental processes.
Which really brings us to a final important question to think about. If AI can now act as this computational microscope to not only elucidate but also reprogram molecular biology, what other fundamental biological processes, ones we've always thought were too complex, too dynamic, maybe too dark to really decipher, what else might now be open to this kind of intervention, to potential rewiring in the coming years?
Yeah.
Beyond just molecular interactions, what's the next frontier for AI in truly understanding and maybe even manipulating life at its most basic level?
Definitely something to chew on until our next deep dive. Thanks for joining us today.
My pleasure.
And we look forward to exploring more fascinating insights with you soon.